Good afternoon, students. I'm back. So today we are going to study the topics, that is development of the male gametophyte and development of the female gametophyte. Yesterday we have generally studied the topic called as pollen grain and also structure of an anther. So pollen grain is generally best defined as a partially developed male gametophyte. It means that for the development of the male gametophyte, the first initial stage is nothing but pollen grain, right? So let's have a talk about development of the male gametophyte, right? The development of the male gametophyte generally takes place from a single pollen grain or a microspore cell, right? So pollen grain is the first initial cell which generally plays an important role for the formation of the male gametophyte. The development of the male gametophyte is endosporic and monosporic, right? When I talk about endosporic means it generally takes place inside the spore, right? And monosporic, it means that it generally takes place from a single spore, right? Okay, so now let's divide this topic into the two part, that is the development of the male gametophyte initiates into two important steps. Number one, that is before pollination in the pollen sac. Number two, that is nothing but after pollination on the stigma, right? So before pollination, the protoplasm, you can see the protoplasm or the protoplast of the pollen grain undergoes mitosis cell division to generally form two unequal cells, right? Those two unequal cells are generally called generative cell and the tube cell nucleus. The smaller cell is generally called as the generative cell and the larger cell is generally called as the tube cell, right? Generative cell generally contains a large nucleus, right? Thin cytoplasm but has no food and vacuole. When you talk about the tube cell generally has a large nucleus, thick cytoplasm and contains the reserved food and vacuole. Now, initially when you talk about the pollen grain, isn't it? At the maturity, the pollen grain is generally two cell state structure during the dehiscence of anther. So we all know what is dehiscence. Dehiscence is a process where the anther generally ruptures to release the pollen grain, right? So during that dehiscence of the anther, the pollen grain is usually two cell stage structure, right? Now the after pollination, now what will happen is that the pollen grain will get deposited on the stigma of the flower and start absorbing a sugary compound called as pectocellulose. So we know that the stigma is sticky in nature because it generally contains a sugary compound. The name of the sugary compound is pectocellulose, right? Now what happens when the pollen grain gets deposited on the stigma of the flower? The volume of the cytoplasm increases in the pollen grain. You can see this is the cytoplasm. So the volume of the cytoplasm generally increases where? It generally increases in the pollen grain, due to which it creates a pressure on the intine, right? As the pressure is created on the intine generally ruptures from the germpore and leads to the formation of the pollen tube, right? So what happens is that the germpore generally gets ruptured and there is a formation of the pollen tube. Now the generative cell, which is the smaller cell, undergoes mitosis cell division to lead to formation of two male gametes. It leads to the formation of the two male gametes. Also the tube cell nucleus enters into the pollen tube along with the cytoplasm. So I can now say that the structure or the pollen tube containing two male gametes, tube cell nucleus along with the cytoplasm is generally called as male gametophyte. So the male gametophyte formation takes place with the help of a pollen grain where the pollen tube is going to contain two male gametes, tube cell nucleus along with cytoplasm is generally called as male gametophyte. Let's have a talk about the second, that is generally called as female gametophyte. Now you know very well female gametophyte development generally takes place with the help of the ovule, right? Ovule is an egg containing apparatus. It means the ovule is generally going to contain the egg, right? Okay, so in a young ovule, when you generally talk about a young ovule, right? In a young ovule, the hypodermal cells which are generally present below, right? They generally differentiates to form the archisporial cell, and one of the archisporial cell during the maturity leads to the formation of megaspore mother cell, which is diploid in nature, right? It leads to the formation of a megaspore mother cell which is diploid in nature. Now this megaspore mother cell undergoes meiosis cell division to form tetrad megaspore, right? This is generally called tetrad megaspore. You can see 1, 2, 3 and 4, right? Now out of this 4 tetrad megaspore, right, the lower one is generally called as the functional megaspore, correct? And the upper 3 are generally called as the non-functional megaspore, right? So the lower one, functional megaspore, the upper 3 non-functional megaspore. Now this functional megaspore will undergo growth. It will enlarge in size. It will undergo growth and it will enlarge in size, and further will undergo mitosis, cell division. So the functional megaspore will enlarge in size, it will gain the nourishment, right? It will grow and it will undergo repeatedly mitosis, cell division to form your 2 daughter nuclei, 4 daughter nuclei and 8 daughter nuclei respectively, right? So functional megaspore undergoes repetitive mitosis, cell division. It leads to the formation of 2 daughter nuclei, 4 daughter nuclei and total 8 daughter nuclei present in a embryo sac. Now this upper 3 non-functional megaspore undergoes degeneration, and that's why they are generally called as degenerative megaspore, right? Now you can see that a embryo sac is generally formed having total 8 daughter cell. Having total how many daughter nuclei or the daughter cell? That is nothing but 8, right? Okay, that is 4 present in the upper pole and 4 daughter nuclei present at the lower pole. Now one of the daughter nuclei from the upper pole and one of the daughter nuclei from the lower pole will come at the center, will fuse with each other to form secondary nucleus, right? So this daughter nuclei from the upper pole, this daughter nuclei from the lower pole, they will come in the center, they will fuse with each other and they will lead to the formation of a secondary nucleus which is diploid in nature. That is, double the number of chromosomes are present in the secondary nucleus. Now, how many daughter nuclei are present at the upper pole? 3. How many daughter nuclei are present at the lower pole? 3. So the daughter nuclei present at the chalazal end will lead to the formation of antipodal cell, right? Okay, and the 3 daughter nuclei which are generally present at the lower micropylar end will lead to the formation of egg apparatus, right? So 3 daughter nuclei present at the chalazal end will lead to the formation of antipodal cell, and 3 daughter nuclei present at the lower micropylar end leads to the formation of egg apparatus. Now, these antipodal cells are generally degenerative. Once the fertilization takes place, they undergo, that is, they generally undergo degeneration, and that's why they are generally called as degenerative cell, right? Okay, secondary nucleus is diploid in nature, right? Okay, and the third one is nothing but the egg apparatus. Now inside the egg apparatus, the central cell is generally called as the egg cell, and the 2 flanked cells are generally called as the synergid. So the central cell is called as the egg cell and the 2 flanked cells are generally called as the synergid cell. So now the structure containing 3 components, that is, first antipodal cell, second secondary nucleus, and third egg apparatus along with the cytoplasm together is called as embryo sac or the female gametophyte. The structure generally containing antipodal cells, secondary nucleus, egg apparatus together is generally called as embryo sac or the female gametophyte. Correct? Now, this embryo sac or the female gametophyte is 7-celled, 8-nucleated structure. Correct? That is, total 7 cells are present. 1, 2, 3, 4, 5, 6. Correct now? And 7. So 7 cells are present and total 8 nucleus are present. 1, 2, 3, 4, 5, 6, 7, 8. So it is a 7-celled, 8-nucleated structure. Such kind of embryo sac is generally called polygonal type of embryo sac. Okay, the next topic is structure of an anatrophous ovule. Okay, so let's first try to understand what is anatrophous ovule. Anatrophous ovule is a completely inverted ovule turned back 180 degree on its top, right? So you can see this ovule is completely inverted. Correct? It is totally completely inverted ovule, which is generally making a 180 degree angle on its top. That is generally a anatrophous ovule. Now, when you talk about the anatrophous ovule is an ovule which is generally present in a angiospermic plant. Angiospermic plants are the plants which are generally highly flowering plants, right? So let's have a structure of anatrophous ovule. Okay, so anatrophous ovule, before that, what is ovule? Ovule is an egg containing apparatus, correct? Egg containing apparatus means ovule is a structure which generally contains a egg, which is a female gamete, right? Now when you talk about this anatrophous ovule, right, it is basically made up of two important parts. One is generally called as the stalk of the ovule, and second is generally called as the body of the ovule. First part is generally stalk. Stalk is also called as the funicle. It is a type of a point where the ovule is attached to the placenta of the ovary, correct? This is the ovule, and ovule gets attached to the placenta of the ovary by a stalk. That stalk is generally called as the funicle, right? Second, it is said that wherever the point of attachment of the ovule to the funicle is generally called as the hilum or the hilus, right? Okay, if the funicle is present in a ovule, it is generally called as funiculated ovule, and if the funicle is absent, then it is generally called as non-funiculated or sessile ovule, right? So the function of the funicle is nothing but it generally provides the mechanical support to the entire ovule. That is, the entire part or the body of the ovule maintains its posture, maintains its support only on the funicle. So funicle generally provides mechanical support to the ovule. Come on to the second part, which is generally called as the body of the ovule, right? Body of the ovule is made up of following important parts. The first part is generally called as the nucellus. Nucellus is a major central part of the ovule. It is generally made up of diploid parenchymatous cell, right? Nucellus generally contains your female gametophyte or the embryo sac. This is the embryo sac or a female gametophyte which is enclosed in the nucellus, right? Now, the function of the nucellus. Nucellus generally provides nourishment to the developing embryo sac, right? That's why nucellus is nutritive in function. Second part is generally called as integuments, right? Integuments are the outermost protective covering of the nucellus. I mean to say is that the nucellus is generally bounded or covered by our outermost protective covering, which is generally called integument. There are two integuments. One is called outer integument and second is called inner integument. Outer integument and inner integument are generally present around the nucellus. The function of integument. After fertilization, the integuments develops into the seed coat. That is, outer integument gets condensed to form testa and inner integument gets condensed to form tegmen. Third part is nothing but chalazal end or chalaza. Chalazal end or chalaza is the basal end of the ovule, and this plays an important role in the formation of the integument. So the function of the chalazal end is it plays an important role in the formation of the integuments, which are the protective covering seen around the nucellus. Fourth one is generally micropyle, right? So at the terminal end of the integument, right? At the terminal end of the integument, there is a small narrower opening or a passage. That small narrower opening is generally called as the micropyle, right? So micropyle is a narrower opening which is generally present at the terminal end of the integument, and the function of the micropyle is that it generally provides the entry for the pollen tube carrying male gamete towards the female gamete. That is the function of the micropyle. Last, that is nothing but your embryo sac, which is also called as the female gametophyte, right? So embryo sac or the female gametophyte is generally made up of three components, right? The first one is generally called as the antipodal cell, second is generally called as secondary nucleus, and third is generally called as the egg apparatus. Antipodal cells are haploid in nature. Antipodal cells are formed at the chalazal end, and this antipodal cell after fertilization undergoes degeneration. So the function of the antipodal cell is they are degenerative cell, as after fertilization they undergo degeneration. Second is secondary nucleus. Secondary nucleus is generally formed by two haploid daughter nuclei. They are diploid in condition. Function: after fertilization, secondary nucleus develops into the endosperm, which is triploid in nature. Third one is generally called as the egg apparatus. In the egg apparatus, the central cell is generally called as the egg cell, and the two flanked cells are generally called as the synelchids. egg cell and the synergids are haploid in nature. After fertilization, the egg cell undergoes formation for the zygote, and the synergid cells after fertilization undergo degeneration, right? So this is the structure of the entire body of the ovule, which is made up of following part: nucellus, integument, chalazal end, micropyle, and the embryo sac or the female gametophyte, which is 7-celled 8 nucleated structure. Thank you.